Yes, sir. You're live now. You can start.
All right. Good morning, everybody. Welcome to all the MOOC students, participants who are taking our courses on bioengineering and interactomics. What we thought, to make this live session more not only interactive but also stimulating for you by showing you where we work, what research we do, different type of technology which we are teaching you in the course, how they are being utilized in our own research, and especially try to give you a different perspective, different context for using various type of technologies. So especially the bioengineering students who are more at the undergrad level, you are exposed to some very basic concepts. Like, you just started looking at the traditional knowledge on the molecular biology and few techniques which are advancing now. But I think what your goal will be in the future, uh, developing towards your biotechnology career or bioengineering career, how some of these high-end instrumentation, software, data analysis can lead to solve some real-life problem, and also, of course, can give you more career opportunities, right? So, therefore, rather than talking again about the genetics and Mendel's law, we thought, why not we choose something which is giving you a little bit more, the broader perspective of the field. So today we are going to talk about clinical proteomics workflow and how that can be utilized for different research, but not only clinical proteomics. We thought to broaden that with the multi-omics aspect of it, which we can also include some metabolomics, lipidomics, various type of technology which we use LC-MS/MS-based workflows to not only address the broad problem, but also some very fundamental problems and biological questions, especially looking at the disease like cancer and looking at finding the druggable target. So we thought to make a kind of theme around that. Again, the students who are joining for Interactomics course, you are studying different technologies to study the biomolecular interactions. And LC-MS/MS is one of the key technologies emerging because you can, with the pull down assays and different other ways of interactomic analysis, you are able to identify the potential interactors, and you can even name them based on the LC-MS/MS based mass spec based approaches. So I'm going to take you to the lab and going to give you the broad view of what we do, various type of instruments, what we are utilizing in our research. And then we have also with us our TAs here. We have Amrita. She's almost finishing her PhD. She has been very experienced TA for the bioengineering and biotechnology related courses at IIT Bombay. And she's your MOOC TA as well. Amit is another PhD student who is looking at the Interactomics course. And then we have Ankit, who is the PMRF fellow looking at the Interactomics course. So I'll first ask Amrita and Amit and then Ankit to introduce themselves briefly to the audience here, because it's the first live session which we are all having. And then I will take you to the lab. So Amrita, please turn on your video and talk briefly about your area of research, what you do, and about your experience for this course.
Hello, participants. Good morning and welcome to the first live session of MOOC Bioengineering and Interactomic course. So I am Amrita Mukherjee, as Professor mentioned. I am working in identifying the critical biomarkers in cisplatin resistance of cervical cancer. My work basically includes the techniques involving multi-omic technologies, proteomics, metabolomics, and some advanced levels of proteomics. So I have been the TA for this bioengineering course for last 4 years, and this year also I will be your TA for the MOOC bioengineering course, and I'll be conducting the next 2 live sessions with you as well. Feel free to ask questions in our portal, and we'll be interacting with you in multiple sessions. Thank you.
All right. Thank you, Amrita. Amit, now you can talk about your work.
Hi all. Myself Amit, and I am a second year PhD scholar, and I'm also working under Professor Sanjeeva Srivastava. So my work is primarily on brain tumors, pituitary tumors. So there I am currently working on rare brain tumors like acromegaly, NFP, and Cushing. And I'm using multi-omic approaches like proteomics, metabolomics, and lipidomics approaches using mass spectrometry-based complex data analysis protocol to understand the pituitary pathobiology and also to find different important biomarkers. So this is all about my project. We have another here, Ankit Haldar.
All right, thank you, Amit. Now, Ankit, you can talk about your work briefly.
Hello everyone. So I am Ankit and I am a PMRF fellow and working as a final year PhD scholar at IIT Bombay. So I had been the TA for many courses and now I'll be serving as a PMRF TA for the Interactomics courses and I'll be taking the live session for the doubt clearing sessions for Interactomics this semester. Yeah. Thank you all and welcome to all the NPTEL members.
All right, so thank you, Ankit, and all the three TAs who interacted in this session. So I think, while MOOC courses, one of the forum which is more virtual, Amrita, if I may request, can you look for Srijita and, I think Srijita at least right now in the lab. She is one of the MOOC topper. And let's see like if she is there. So again, students, what we are looking at here, that despite being virtual forum, virtual platform, we do feel like, the MOOC offers us the level of connectivity, which makes us feel like, okay, you are not just virtual, in different places. We are all interacting with you live. And every week, of course, you watch our videos, but also we give you the live session for the Q&A every week, forum-based queries. But we don't end our journey with this. The students who take our courses and perform very well, we do want to do much beyond that for them. And despite that, at IIT Bombay, our labs are very busy and a lot of researcher students are working in our lab. We do give opportunity to the topper students, the best students of the MOOC courses to come to our lab and do some training. And I think Srijita Ganguly, she is one of the students who was the topper of Proteogenomics course in last semester. She has just currently joined our lab for her internship program. And two more students are just joining. And I'd also like to thank the NPTEL team who provides them some fellowship and support and facilitate, coming to IIT Bombay. But again, like as I said, our journey does not stop with, some video course and just talking to you, the virtually. But we truly believe that this gives us a forum to interact more. And if you are the good learner, we are there to help you and take this journey forward with you. So Srijita, if you can just, talk briefly about your experience of MOOC courses and now coming to IIT Bombay as an intern.
Thank you, sir. I did the NPTEL course on the Introduction to Proteogenomics for the 12-week course, and it was a amazing experience for me in online because every details and every concepts were, very simplistic way and a very, interesting way they were explained. But I didn't expect that I will be here in this lab and join us in person and, be a part of this lab. So it's really amazing. And thank you so much to NPTEL and to Professor, of course, for giving me this opportunity.
Thank you, Shrita. Thank you very much. But also let me say, like when I looked at your performance and your score, you did so well in a very tough course like proteogenomics. I thought you have to come to our lab. Okay, so when I look at every student's performance finally, these are sometimes the difficulty level of this course is not very easy. And we do try to keep it, the questions in the final exams are similar to IIT Bombay. So therefore, you perform almost 90%. And I thought that, you deserve to be here despite, as you can see, lab is very busy with a lot of people around here. So again, students, like, this gives you probably motivation that we are not just going to stop our journey with these courses, but we will be happy to take your learning forward. We're also going to conduct one international conference and workshop at IIT Bombay. And briefly, let me just show you before I will probably forget about, this thing in the live session. Let me share my screen and try to show you this event. This could be another opportunity for the students who want to interact more and try to get some knowledge from us with the training program. So we are going to conduct almost 12 workshops in February, 14 to 17 February, the Advances in Proteomics Technology APT, with the workshop, as well as we are going to have the 2 days conference. So this gives you the opportunity to also come to IIT Bombay as a part of this event. And again, later on, if you perform very well in the course, you can be given opportunity to come and do the longer training and be part of the internship programs. All right, I hope that gives us some level of interaction. Let me now go to the lab and just give you briefly about our lab overview, what we do, and then we have made some plan how we are going to show you some workflows in the proteomics, metabolomics, and multiomics. All right, I'll just take a minute to reach to the lab, and then I'm going to talk to you briefly about our research work. Yeah, okay. All right, so I changed my dress. I am now in the lab coat because I'm in the lab area for biosafety. So welcome to the high resolution mass spectrometry national proteomics facility here at IIT Bombay, which we have established. And variety of things which we talk in the advanced technology and tools for the bioengineering or in interactomics. Many of these technology remains quite relevant to you. And we thought to give you some exposure of it. We are using various type of LC-MS/MS based platforms. So what you see one of the instrument in my back is the mass spectrometers. And there are a variety of mass spectrometers which can be utilized for different applications. So one of mass spec which we are showing you right now is known as the Orbitrap fusion. And this particular mass spectrometer can be utilized for deep proteome analysis. It means if you have a complex sample where you have thousands of protein and you want to screen those protein, you can utilize this instrument. If your goal was to quantify the two different conditions and want to look at differential responses, again, you can use this technology. Any of the mass spec has usually a chromatography component. In this case, we have the nano-LC component. And then we have the front end. The first part is the electrospray ionization, how you ionize your peptide to become ionized. And you can see the insert of it, where the ions are going to move inside the mass analyzer. This instrument has three different type of mass analyzer, known as the tri-bit technology, having the quadrupole, Orbitrap, and ion trap. So this instrument we mainly use for the deep proteome and quantitative proteome analysis. Another instrument which is on the side, another LC-MS/MS, where we have the UHPLC chromatography. And again, we have the electrospray ionization and a hybrid type of mass spectrometer, which has the quantitative mass spec containing the quadrupole and Orbitrap. This one we mainly use for the various type of metabolomics application with the high flow rate. So again, LC-MS/MS could be utilized for different type of application for that matter. Now on this side is another technology which is the triple quadrupole mass spec. Once you have done the different ways of discovery work, now you want to validate your target, whether it's the proteins or metabolites or lipids. You are now knowing the specific molecule of interest. So you don't have to screen again for hours and hours. So now we can do the targeted proteomics or targeted metabolomics, again with the chromatography with the high flow rate. And then we have the ESI, and then we have the mass spec, which is a triple quadrupole mass spec, which can be utilized to screen the specific set of our metabolites or the proteins and peptides. This just gives you some flavor of the different tools available in the lab, which we utilize for different workflow in the clinical proteomics and multiomics area. And the technology which could be also of interest is this instrument, which is known as the BLI or the biolayer interferometry. Here we have the biosensors, and then we have the plate where you can keep a sample. And what we are looking at the protein-protein or the protein-drug interaction analysis. So you're looking at the biolayer interferometry technique to monitor the physical changes across the two different molecules. So again, this could be done in high throughput manner, and one could use that way to screen for the proteome. So I hope you are getting the broad sense of it. These are the very important gadgets, important tools, and state-of-the-art technologies, which are quite important for us to utilize in different biological problems. So my lab primarily does two type of major projects, one on the area of the brain tumors, especially on the gliomas, meningiomas, pituitary tumor, medulloblastoma, and variety of other cancer types. especially we started some cancer moonshot project where we are part of the oral cancer, cervical, and breast cancer as well. So we look for the variety of sample type from the serum, plasma, tissue, saliva, cerebrospinal fluid, and every type of biological sample comes with its own challenge. Idea is how to get the thousands of protein out from those samples, analyze the healthy versus disease sample, and then try to find out the intensity level of these and look at the pattern analysis. Can we find out some pattern of the healthy versus disease, and then use the different ways of statistics followed by machine learning to narrow down these leads, and then see what kind of biological information they have and inference they have, right? So again, these tools help us to narrow down those leads, and then we do a lot of fundamental biological questions to address, especially looking at the different cell culture model and various other type of technology to understand those pathways and biological information from them. So I hope that gives you at least, a bit noisy, but the feel of the real lab, how the things actually work in the real lab. But from here onwards, I'm going to now take to our TAs who will discuss with you some specific workflow. I'll now invite Amrita from our team to talk to you about a specific technology known as cell surface proteomics. Again, if you were to look at the cell, you have all the globular protein, all the protein come from the global protein analysis, but they are very abundant. If you're looking at the druggable target on the membrane on the cell surface, so probably you need to only enrich the cell surface membrane protein and not the content from inside. They don't lie so rigorously in that. So Amrita is going to now talk to you about the cell surface proteomics workflow and how one could utilize these tools what I'm just showing to investigate some specific interesting clinical problems in the proteomics world of personalized medicine. So with this, over to you, Amrita, for taking the students forward for this workflow on the cell surface proteomics.
Thank you, Professor. I hope I am audible and the screen is visible. So, and today's talk is mostly about the clinical proteomics. And when we talk about clinical proteomics, we always think about to build personalized medicine. And towards building personalized medicine, a specific type of omics technologies we use, and which is called targeting the cell specific surface markers. Now, if you for example, if you want to examine any biological or clinical sample, for example, blood, and this will have various cell types, right? Blood will have various types of cells, starting from the red blood cells to the several types of white blood cells and others. And how do you want to differentiate them? How do you want to know what are the various cell types? You would definitely go for a microscopy. So under microscope, you'll be able to see different types, depending on the different cell types, you'll be able to tell that these are the several various types of cells. But what if some of the cells look exactly the same? For example, the subset. For example, in the blood, we have the T cells, and there's various types of T cells like regulatory T cells, then gamma cells, then T helper cells, and other types of T cells. They look exactly the same, and using the microscopic techniques, you won't be able to differentiate which cell is what. Then what you would like to do? Then you would need for the biomolecular markers. So the biomolecules that are present on the surface of the cells, they are the major identifiers of the various cell types. Now, let's look at what does the surface of a cell look like, how we can actually differentiate from one cell to another depending on the biomolecular signature of the cell surface. So a typical cell surface looks like this. It will have certain types of proteins, it will have the lipids, it will have the carbohydrate molecules protruding out from the cells. So the bilayer of lipid and intruding the proteins. Also from the proteins, there will have the single pass proteins, there will be the multi-pass proteins, there will be peripheral proteins, that there will be proteins protruding out of the surface, there will be proteins that are emerged on the bilayer. So these are the basically the surface of a cell will look like. Now, the question arises,
what does the surface protein, what do they do basically? So surface markers or the surface proteins are mainly involved in various types of cell activities, specifically for the signaling pathway. So whenever a signal comes from outside, there are the receptors. Surface receptors are always there to take up the signals, and depending on the signals, the downstream changes that happen in the inside the cell, which will decide the fate of the cell, whether the cell is going to divide, whether the cell is going to have replication, or whether the cell will go to apoptotic mode. So everything, whether the cell will differentiate or not. Other than that, this cell surface molecules, they are not only the receptors, there are the interactor proteins. So in immune system, a lot of cells interact with each other to pass the signaling, and the cell interactions are mediated mostly by this surface proteins. Then there are adhesion proteins. If a cell want to adhere to some sort of new tissue site, and this basically happens during the disease progression and in cancer or metastasis, where the cell basically the cancer cell basically detaches from the site of its origin and then go far and then go into new site. And of course the immune reaction. So basically, the surface proteins are involved in multiple types of cell-mediated activities, which make them very important candidates to study, right? Then again, why they are important? Because this surface markers or the surface proteins are often called as surface markers. So they are, because they present on the top of the cell, and that's why they are very much accessible to antibodies, so that they can be tagged. They are accessible to drugs, so that if you are, first imagine a disease condition where a particular disease cell has a type of surface protein, and you want to target that cell and treat that cell or kill that cell with a particular drug, so that is accessible. Also, these surface markers are highly specific to certain cell types or disease states that makes them a very ideal for targeted therapies. So basically, they help us in better diagnosis. The surface markers are they help us in building enhanced therapeutics. All right. Now, in this talk, we are going to, this session, we're going to talk about how to study all the surface markers at a go. So, the cell surface proteomics, it's a technology, advanced technology that help us to study all the surface protein, all the surface protein that are present on the surface of a particular cell at one go, rather than going and studying them one by one. So today we are going to talk about, there are many methods, but one of the most famous method is N-glycosylated cell surface capture technique, where what we do is that we take the cells and we tag the cells with biotin. So basically, and then we pull down the entire thing and we do the analysis. We'll go the steps through one by one. For suspension cells, the technique is different. For adherent or epithelial cells, the extraction procedure is slightly different. So now, capturing the N-glycosylated proteins from the surface of the cells, how do we do that? So for example, we start with the adherent cell, and then what we do is that we biotinylate the N-glycosylated proteins, which is N-glycosylated proteins majorly they are the more than 80% of the surface proteins are N-glycosylated. So this the carbohydrate moieties are biotinylated. So all of the proteins that are on the surface of a cell, so basically they are tagged by this biotin, right? Then we lyse the cell. Now, when we lyse the cell, we'll have the cell broken open, but in the solution, only the proteins that are outside of the cell, and some of the proteins that are secretory proteins and other proteins, and some few fraction of the proteins will be biotinylated, right? And other intercellular proteins will not be biotinylated. So here I'm showing how we have biotinylated and snap frozen the cell pellet. At this point of experiment, you can freeze that particular pellet for a longer period of time in -80°C or in liquid nitrogen for future use. Then what we do is that we take the lysate, which contains both biotinylated proteins and the non-tagged proteins, and pass it through a column, which will have the neutravectin, and the biotin reacts with the neutravectin, and only the proteins are pulled down that are tagged, and that's how we enrich the cell surface proteins. Now, I'm showing here how we have done it. We regularly practice in our lab. This is a particular column. As you can see in this particular column, you see the gray area. These are the neutravectin beads, and they pass through the column, and we incubate the neutravectin beads with the biotinylated cell proteins, and at the same time, when they are captured on the bead, we do the tryptic digestion to make them in peptides, because we are going to run those in the mass spec, and in mass spec we cannot run the entire protein, right? We have to first digest into the peptides, and then we load the peptides into the mass spec, and then the mass spec run is complete. Now, once you identify the pull down the peptides, you run it on MS, and then you collect the raw data, and then the raw data is analyzed through several softwares like FactPy, like Diet, and taking the raw data, you would like to know what kind of surface proteins are there, right? For that purpose, you use a database called SURFI. This database is built for the surfaceome mapping. Let me show you a little bit of how does the website look like. I can show you the so if this link opens, I'd be able to show you how you can actually visibly show the database. So it's a database built by Professor Bern Goldscheid and his team. Yes, this is the interface of the website. So it has more than 2,500 surface proteins that have been identified, and taking those, it has been built. And if you start exploring the surfaceome, whatever data you have got, you can load your data and run the analysis over here. And this entire image shows the currently available surface tree map. If you go closer, you will be able to see in each domain all the identified proteins are mapped. So color coding identifies different types of surface proteins. For example, the pink R1, the transporter, they have been identified so far. The blue ones are the receptors, they have been identified so far, and so on. So basically, and then you basically map your raw data with this interface, and you identify what kind of proteins you have got from your study. So this is a particular image that shows that what are the surface protein types that have been generated, taking a total of 610 cell lines, and the color code color code tells you about that particular tissue type or the cell type, what are the number of surface proteins we have got so far. So this particular website has identified 2,800 high confidence surface proteins that belong to various types of categories. Apart from that, there is one more technology. It's called LuxMS technology. This technology will enable you to identify not only the surface proteins at one go, but also the spatial surface interactions. For example, how different types of proteins are interact in a very nanoscale manner to to maintain the specific properties of cell surface or maintaining during, and what changes at what millisecond changes happen during one type of cell signaling or receptor ligand interaction, etc., etc. Now let me talk about briefly talk about one very real world example of what we have done in our lab. So I have tried, as I said, that I work on cervical cancer, cisplatin resistance. So I wanted to know what are the surfacium, how the surfacium look for the HPV positive cervical cancer cells for both the cisplatin sensitive cells and for the cisplatin resistant cells. So idea was to identify the unique markers present on the cisplatin resistant cells of HPV positive cervical cancers. So here I have in my lab, I have prepared a cisplatin resistant cervical cancer cell line, and as you can see, this is a typically resistant cell look like. They are larger in size, but from here I cannot know, right, what are the surface, how does the molecular at molecular terms how the surface look like. So I have taken those cells and I have tried to I have done the cell surface extraction from sensitive versus resistant cells, and I was able to identify more than 150 differentially expressed surface proteins. These are which I can call the classifiers of cisplatin resistance in cervical cancers, and they belong to various categories, and that cell addition migration, some belong to cell signaling, some belong to the growth receptors, other are transporters and receptor threonine kinases, all types of markers that are differentially expressed in two types of cells were identified. Now, with this technology, we also, a small fraction, we also were able to identify non-surface protein members. For example, the membrane proteins of lysosome, the peroxisome, nucleus, Golgi, mitochondria, etc., etc. And we were able to get a lot of endoplasmic reticular membrane proteins as well. This is how they look like. For example, for the mitochondrial membrane proteins, we got the outer membrane proteins, inner membrane proteins, and intramembrane proteins as well. So this technique is a very useful technique that can be used for clinical proteomics to translate. And it has huge translational abilities because the markers we get from this type of omic study that will classify disease versus healthy state, and that can be taken forward to build very promising therapeutics, because already I told that surface markers are really unique, and that's how it is. Thank you, everyone. Now I pass the talk to Mr. Amit, who is going to talk about various other techniques like metabolomics and lipidomics and how they are used in clinical omics platform. Thank you.
Hi all, think I'm audible. Hello. Hi all, I think I am audible. So today, as Amrita has just said about surface proteomics, and today I'm going to also say about how to process metabolomics and lipidomics samples from serum. So basically our lab is not just only subjected to proteomics. We are also exploring different small molecules using complex metabolomics and lipidomics workflows. So today we are going to show that how we can separate the metabolites and the lipids from the serum samples. Okay. So first, coming to metabolomics. So basically in metabolomics we use 100% of methanol, and in case of lipidomics we use chloroquine and methanol mixture in 2:1 ratio. So in the very first stage, when we have collected the blood, we need to separate the serum from the blood. Okay, so that's why we use a specialized tube to separate the serum. We basically run it in a swing bucket, in a centrifuge. We've got a swing bucket. we run it in 3000 RPM in 4 degrees Celsius, and in the upper part you can see there's a yellowish liquid there. It is called the serum. So we take the serum and we give 100% methanol in 1:4 ratio in a cold temperature. So basically the objective of using methanol is that it basically precipitates the proteins and all. It is, and we also keep this in a very cold environment because it will give a cold shock to the sample. So otherwise there will be other enzymes that will get activated and that will change the metabolomic profile in the samples. So in case of, after using 100% methanol, we need to incubate it for 8 hours long, and after that we need to use a centrifuge and we to run it for 14,000 G for 10 minutes. After that, we will get a supernatant. From supernatant, we need to dry the supernatant. Then we get the metabolites. Then we use the QE mass spectrometry in our lab to see what are the different metabolites are coming. So before analyzing the before giving it to the mass spectrometry, we first need to use the PTV filter. So that basically is important to filter out the samples. Then we'll go for statistical and pathway-based analysis, where we use different commercial softwares or free software to make sense of the data. Similarly, in case of lipidomics, we use chloroform methanol. So basically, it will make a biphasic biphasic layer. So whenever we use chloroform methanol, the chloroform will basically distort the hydrogen bond, the electrostatic interaction of the proteins and the metabolites, and the chloroform, their metabolites will be dissolved in the methanol. And in case of the lipids will be highly dissolved in the chloroform. That will be in the lower part of the tube. So similarly, we isolate the serum and we use the chloroform methanol extraction protocol. We use an orbital shaker to mix all the chloroform methanol with the serum sample. Then we incubate and we add. And similarly, we use a centrifuge and we run it in 14,000 G. In RT for 10 minutes to get the metabolites and the lipids from the same sample. So after that sample is dried in the speed bag, and after drying the samples, we need to reconstitute it IPAC and methanol mixture. After reconstitution, we use the PTFE filter to filter out the samples. Then it will be subjected to mass spectrometer based analysis. After that, there will be statistical pathway analysis where I use commercial softwares like Lipid Surge, and there are also pathway-based softwares like Metabolist and all. So right now, I am going to show you that what are the things we generally use in our lab to process the samples for lipidomics and metabolomics. Yes.
So I think it's a good idea that we should also, show a little bit the actual instrument and technology, Amit and team. But again, students, I have shown you the instruments and you can see the possibilities are endless, right? You can utilize these instruments in different modes. You can utilize for different biomolecule. It just depends on what questions you want to address. And from the same sample, you can try to extract out the protein or the metabolite or the lipid. Of course, you can do nucleic acid for genomics and the transcriptomics. So a lot of things can be done. And idea is with this kind of high throughput platform, can we try to get maximum knowledge out of it, right? To really build our hypothesis strong. So I hope this gives you a little bit, the idea, but Amit and Ankit will now take you from the lab and show you some of our setup, plus the data analysis skill in that area. So Amit, you can continue with the QE demonstration now.
When we have group studies, but that's why we collect the blood. This basically specialized for this. So we have basically keep the tubes here, and that's how we keep the tube here, and we run it in the center. So before for isolating, the serum, so we need to keep the temperature for 4 degrees Celsius, and the speed we can keep 3000 RPM or just higher than that, and temperature we need to keep 4 degrees Celsius. So after that, when we ever get the serum, so after getting the serum, we need to for metabolomics, we need this is 100% of methanol to be 3 methanol, and these are the serums. So you can see the yellowish part. So this is the serum actually from where we are going to separate the metabolites and the lipids. So first of all, for the metabolic protocol, we take 100 microliter of serum, and we add 400 microliter. We add 400 microliter of methanol. We mix it, and we need to incubate that for 8 hours in minus 80 degrees Celsius. So now, whenever we do the one night incubation, you will see this kind of white layer or whitish color you will see. So this is basically the serum and the methanol just kept for 8 hours in a minus 80 degrees Celsius, and after that we need to centrifuge it again in 14,000 G 4 degrees Celsius. After that we will get a supernatant, and that supernatant we need to dry in the sweetener. So similarly, in case of you say about lipidomics will also try to take the lipids protocol I just explained. So we can also isolate metabolites and the lipids from the same kind of sample. So similarly, for the lipidomics, we can take 50 microliter of sample. We need to add chloroform and methanol in the 2:1 ratio. After that, we need to mix it in an orbital shaker. So this is basically an orbital shaker. Suppose you will have your mixture here. In the mixture, basically you just need to keep it, and you can just run it for 1 hour. So for 1 hour it will run like that. After 1 hour gets over, you need to add the water. And you'll go for vortex. Okay. So I have already done the protocols. I have already performed the vortex, added the water, and I have also centrifuged it for in 4,000 10 minutes in RT for the lipidomics protocol. So after basically running it in the centrifuge, you can see there are three different distinct layers are coming. I think you can see these three distinct layers are there. So in the lower part, there will be lipids molecules will be there. It basically dissolved in the chloroform. In the middle part, there will be proteins, and in the upper part, there are the polar metabolites, which are dissolved in the methanol. Now, whenever you are going to, suppose from one sample, you are trying to do metabolomics and lipidomics. So similarly, you will take the upper, if you take the upper aliquot or upper layer, you will go for drying. You will get the metabolites there. And if you dry this lower part, you will get the lipids from the sample. So not just that. So after getting the metabolites and the lipids from the samples, it is not just over. So now after whenever we have dried the samples, you can store it in the minus 80 filters. So now whenever suppose you are now going to run it in mass spec, you can store it for a while. But whenever you are going to use the mass spec, as I said, we need to reconstitute all the samples, whether it's from Japanese or it's from Interactomics. So after reconstitution, you need to filter out the sample. So for filtration, this is actually the PTFE filter. If you closely see, so this is the filter you can see. So every time when we are going to process the after processing the sample, when you are going to analyze or you are going to give the sample to mass spec, you need to first pass through the samples through it. Okay, so you can see the diameter 0.22 micrometer. So after it is getting passed through this filter, your samples are ready for the injection, for the data application. So in case of metabolomics, we use this kind of vials. You can see the transparent vials here. In case of lipidomics, we use this kind of amber vials. Okay, so amber vials, you can see it's quite light sensitive actually. And so the reason is that sometimes in lipidomics protocol we use internal standards. Okay, so for internal standards are basically highly sensitive to light. So to keep it nice, we use this kind of amber vials. So and another thing is that so these are the inserts. So whenever you are giving the samples in the mass spec via virus, you need to keep the inserts here. You need to put the samples in a vials and we'll close the cap. Then you can just give this vial into the auto sampler. So I should so let me take you to the mass facility where I can show you that what actually is the sample.
All right. So sometimes sample preps are actually quite easy, right? And then, of course, these instruments are very powerful to generate data. But the major challenge comes into how to analyze the data, right? But let's take the next step now and just show you the LCMSMS, which we have already showed you, but how that can be utilized with the high flow rate for the metabolomics and lipidomic analysis. So Ankit and Amit, over to you.
That it is like 5 mm in a high flow rate in comparison to nano LC or micro LC we use, because in this kind of operations what we need for metabolomics and lipidomics, we need to the analyte to pass the high flow rate and the analyzer. So before going that that nothing, this is the analytical column. This is how it is here your particular journey. So we just keep our samples here. When we keep our samples here particularly, and when we just stop it, and then when we go above, and we just place the command, right? So when we place the command for the sample, take the sample, take our vector volume. So if we take vector, it will put the particular paper where it will be fixed, and then these are our solvent system. So this is ultimate 3000 HPLC, which is consists of there are four pumps. So there are which we generally use like the two solvent A and B. So since we try to make a hydrophobic gradient, so we need different solvents to make a gradient which pass through the column. So when the sample we generally take, and then it comes to the column. So in that it will be the stationary phase. The samples will get bind to this material. So for example, this is the C18 material. So the hydrophobic of the molecule will get bind to the column. But now when we use this while phase and increase our gradient of the hydrophobicity, then our molecule will elute it on the basis of their hydrophobicity. And after that dilution, it will go to our analyzer. So there are two types of analyzer as already known. The two exactly for metabolomics, like we have the quadruple and orbital. So there the MS1 and the MSMS occurs to identify a particular lipid or metabolite. So how it generally happens is that on a typical chromatogram. So this is the total ion chromatogram of all the ions that are generated. After dilution, then there is after it has passed the electrospray ionization. So this is how the ions are detected over the time. Here you can see this is the time, that is the retention time. So this was a 32-minute gradient of different isotopic gradient that has been passed through. So the compounds have been eluted out and have been we have got. So this is a, now you can see that it is written like waiting for activity to start. So it will be a complete live run. So you can see that how this detection will happen and it will start running and analyzing. So when we tend to give this analyzer, so one it performs is the MS1. So that is the complete precursor that it takes. So whatever be the M by Z or molecular weight of the entire metabolite or a lipid, it gets we try to detect, and then a fragmentation occurs to get the MS. So now you can understand that for a particular metabolite or a lipid, same molecular weight metabolites and lipids are present. There are many metabolites or lipids that have the same molecular weight, but their MS fragmentation pattern is what that will be unique, and that's where we come. That where we can try to identify the particular metabolite or lipid. So if I just show you one thing here, so yeah, yeah. So this is how we tend to keep the gradient of the format. You can see that there is a gradient in the microactivity and then coming back, and this is where we keep on our different method of different resolution and different ionization on the dependence of our scan rates and so now this one, so you can see in the live run when it happens, so here you can trace it out that how your different ions are getting analyzed. So this was the little brief about the instrumentation. So you can see a live run now injection being taken in the from the autosampler, and it will be injected. You can see that it has sample as taken, so it is quite automated way. So you can have your own particular, you can give the position in the computer system and it will take. And here now it is getting fixed and it will now get into the analytical column where it will get bind to it. And see, now you can see in the screen that it has already started erupting and it is started for analyzing. And if we just measure here. So this is how you can see the emission and the fragmentation that is happening, and it is showing in the this kind of high speed. You can see all the ions are getting detected the analyzer in the detector, and we are getting the result. After this, we need the analysis to find out that which particular MYZs or which particular after this even in the software where you will try to access the database that one of MYZs and getting the one and the so all these will be gaps find and identify that that we are getting. So again, just to bring it, so just to show you the entire setup once again. So this is the HPLC going through the column, so which we use like reverse liquid chromatography, followed by ionization source, so which is our electrospray ionization, and then it goes into the analyzer, and we can monitor and find out that. Yeah. This was all for the problem for metabolism.
All right. So I think you got the the visual sense of how one could utilize this platform to analyze the data. Before we talk to you at least some data analysis aspect, let me just take one or two query which was given on the forum. One student asked that I'm a chemical engineer and want to study this process. how the interactomics and biology can help me out in this area. So I'd like to say, like, even at IIT Bombay, we have all the students who come from engineering studies. And the first year student, they are having mandatory 101 course when they study different biological and technology things in the biology area. The reason for that is whether you come from chemical engineering or computer science background, these fundamental questions as well as these technologies are going to be help you out in different steps of decision making. Again, the LCMSML based workflow to look at impurity testing for different compounds, looking at optimization of different chemical processes, all of that it can be utilized. And towards scale up, then when you are going to take the decision as a process engineer, your fundamental knowledge in these areas will be quite helpful. Second question is, can the interactomics be studied using simulations? Interactomics, what you're looking at the interaction of the two biomolecules. You're looking at how do they interact. And first, one need to generate, so it goes in multiple ways. People do a lot of simulation studies because if you're looking at, let's say, protein, whether it can be inhibited with some drug. So you just don't want to start an experiment because you have to grow the cell and you have to, do a lot of experiments and buy the very costly drugs. You don't want to do this kind of experiment to begin with. You first do some simulation study, some docking study, how these small molecule protein they bind and they interact. So first, a lot of computational work goes on to this process, and then one can do the actual experimental work to validate that. So of course, the simulations and the MD simulation and different type of docking studies are very heavily utilized in the field of interactomics. Field of interactomics could be quite helpful to understand the different type of interaction analysis. And again, once we have generated some data, so let's say think about how the two technologies can be matching each other. So for example, the experimental skill and the computational skill. So I was studying one like in one of my previous work, looking at the p53 protein and its interaction with MDM2. This is very biologically relevant protein p53 because that is involved in many cancers, and there are different type of mutation happens for p53. So we generated, let's say, 50 mutants of p53 and wanted to study its interaction with MDM2. So now let's say you generated 50 mutant and you purified each of the 50 mutant and you did experimental study with the MDM2 and look at, is that binding, or because of the mutation the binding is not happening. But how many mutation can you actually study in this manner? So with the 50 mutation panel looking at in different regions of the p53, we were able to generate some data which was able to give us the foundation. But now based on that data, looking at different regions of the p53 structure, one could now think about utilizing that data to do more simulation. And now if I have to look at some unknown mutation happening in the same region, then probably I could utilize my parental previous data and simulate that and try to see at least closest what I can get that interaction data or the interaction value. So a lot is going on in system biology field where people try to generate the experimental data first, use that to build the models, and then use that towards your intelligence and with the machine learning towards making more predictions in the unknowns. I think that's another way of thinking about it. And therefore, irrespective of your background, these knowledge of the bioengineering, the interactomics is going to really be very helpful to you, taking the your career forward. If there's any query in the forum or on the YouTube, TS, please, can you tell me?
So I did not, there's no point in the forum for the interactomics.
Okay, so nevertheless, I think there's a good beginning of interaction towards our MOOC courses. I hope everybody started enjoying the course. And as I mentioned, we are not just doing a virtual course, online course. We are actually quite available to make it for you to feel like in-person course where we are quite accessible to you. Please do send your queries in your forum as well as I'm accessible by email. And then if you are the one who is performing very well, there'll be a lot more incentive, a lot more rewards for you. And we'll look forward to give you more and more opportunities in terms of whether you want to come to some workshop and conference at IIT Bombay, or even if you want to come for the internship programs, or look for some future job opportunity. I have very successful stories from my lab where MOOC toppers have come here. They, in fact, some of them stayed for doing PhD and then went to top universities in Harvard and California and different places for their respective career journey. So it has been very stimulating experience for both of us. I think for the student as a participant and for us as a mentor, it has been quite rewarding journey. And we believe truly that, this is a good learning experience for us. And we look forward to have continued interaction with you. Please do utilize the live sessions and live interaction, because this is a way for you to talk to us more organically towards your career issues, towards your apprehensions, towards your more questions about the fundamentals. All of that we are very accessible. And we have a very good team of the TL, as we already introduced. Amrita, Amit, and Ankit, these are all top students from IT Bombay and quite experienced in their own research. So do utilize to interact with them and try to get the best out of these courses. Good luck, everybody. We'll again meet you after a month in the next live session with new story and with new demonstrations. Okay, till then, bye-bye.